Good morning, everyone, and welcome to the Coffee Talk that's hosted by the Northeast RISC. It's out of control. We're going to be talking about how does climate change affect biocontrol agents and their hosts today. But before we dive into the meat of this topic, I wanted to give a little bit of background both on the RISC network and also just a little bit of an overview on biocontrol so that we're all on the same page going into this. So as for the background on the RISC, it was founded in 2016 based on a request from invasive species managers on how to manage biocontrol invasions, biological invasions in light of climate change. When this question was posed, Bethany Bradley, who's a professor at UMass, a leader of the RISC network, and also co-director of the Northeast CASC, Tony Lamorelli, who's a research ecologist with the Northeast CASC. And at the time, I was at Cornell as director of the New York Invasive Species Research Institute, but today I'm actually the regional administrator of the Northeast CASC. So this question came up, and we were trying to figure out a way to help managers get the information they needed. With that, the Northeast RISC was founded with the mission to reduce the compounding effects of invasive species and climate change by synthesizing relevant science, communicating the needs of managers to researchers, building stronger scientist manager communities, and conducting priority research. Today, we have a big leadership team that meets every week to address these issues and synthesize information and conduct research. And we also have nearly, I think it's around 800 people in our network today as well, managers and researchers who have been a part of our efforts. I'm not going to go into depth on this, but I just wanted to highlight that the RISC focuses on 4 main areas. We work in boundary spanning, so this effort of bringing together researchers and managers and bridging the gap between them. We conduct original research. We translate and synthesize research, and then we focus on communication and implementation. And the process that we use is a translational invasion ecology approach. I think they're going to drop the link to this paper in the chat, but really, starting with our stakeholders to identify the problem and then coming up with ways to address those problems. It's really exciting to me that although we started in the Northeast, now there's RISCs across the US and into Canada that's being established right now. And so if you're calling in from somewhere that's not in the Northeast, just know there may be a RISC in your region that you can join and participate in for regional specific topics that come up. All of us have websites. So here's the website where you can go in and find a link to all your RISCs, including the Northeast RISC. And so also if you are interested in being on the listserv, if you're not already, feel free to go in and sign up so you can get notifications for coffee talks like this one, or when we have any other information coming out. The focus of our talk today is on biocontrol and climate change, and that wasn't just something we randomly picked. Annette Evans and Eva Kohlberg, who are on this call today and who you just are hearing from now, conducted a survey. This is the second survey, so we did one 5 years ago. We did another one recently, and the efficacy of biocontrol under changing climate came up based on managers' responses as a top priority and a research need, which is what really drove us to dive into this topic. Quickly, I know many of you are on this call because you already know about biocontrol and you're just interested in the impacts of climate change on it, but just so that we're all talking about the same thing, I have a couple of slides on biocontrol. So what is it? It's the intentional use of one living organism to control and suppress another organism, such as an invasive plant. And there's all different types of organisms we use as biocontrol agents. And I just want to acknowledge that these slides, many of them are based on slides of my colleague and friend Jen Andres, who's hopefully on this call. I can't see the list of participants right now, but just to make sure she gets credit for all of her great information on biocontrol that she shares with us. There's 4 types of biocontrol. Augmentative biocontrol, which is just trying to augment naturally occurring populations. Conservation biocontrol is manipulations of natural enemy populations to favor the natural enemy. Cultural biocontrol, such as utilizing other herbivores such as goats. And then classical biocontrol, which is the introduction of natural enemies to control and introduce pests. And that's the one that we're really going to be honing in on for the purpose of today's talks. It's based on the idea that in a native range, both there's pests and then there's natural enemies there. But when we move into the introduced range of a species, a lot of times those natural predators are not present. And so we need to purposefully introduce them. And this is based on the enemy release hypothesis. Oh, we've got lots of, and how it works really is that the host plant or target species, whether it's an insect, is present. And then when the biocontrol is released, it increases to prey on that host. And then as their populations go down based on the predation, also the biocontrol agent goes down. So we won't have these out of control populations of biocontrol agents. Really just pointing out that when we use this strategy is in this red zone when species are already widespread and difficult to manage. We use biocontrol in places like agricultural settings for crops and in orchards. We use them in natural rangelands, in urban settings such as greenhouse crops and in landscaping, and also for human health purposes. So how do we consider a biocontrol agent being successful? Well, first of all, they have to go through a long approval process, which I don't have time to go into today. But for them to be approved, they need to feed on the target organism and impact it in the lab. So before they give it the okay to be released in the US, it has to meet these standards. But then also for a biocontrol program to be deemed successful, these agents have to the biocontrol agents have to establish in the introduced range and decrease the population of a target organism such as swallowwort or any other species that we're trying to reduce, and then ultimately benefit the native species and ecosystems by reducing those populations of the invasive or pest organism. There's a lot of advantages to using biocontrol. It's ecologically desirable because it doesn't have non-target effects. They move around, they move and expand their range, so it's not the impact and the success doesn't just happen at the point of release, but expands beyond that as the population grows and spreads. It's a long-term solution. Once you get established biocontrols, they are there and they can continue to do their work without further intervention. It's gradual, so we're not just clearing out a landscape. It gives a chance for the native organisms or plants in many cases to fill back in. It's very cost effective in compared to other control techniques. And it's safe. We really biocontrol has gotten a bad name over the years. But nowadays there's such a stringent approval process to make sure that there's no effects outside of the target organism. However, there are some challenges to biocontrol. Is sometimes not compatible with other management techniques. There may be predators that are in the natural range that are feeding on those biocontrol and thus reducing them. There is a risk of non-target effects of non-specific biocontrol agents, although those aren't used anymore because of their non-specific habits. There is a possibility for mismatch in phenology between target and biocontrol agents. So maybe one is blooming and out and the other one hasn't come out yet in the spring. So that's a challenge as they're introduced into a new into a new place. Their ranges might not overlap. And also natural disasters like fire that's shown here can reduce a biocontrols population or wipe it out. And so that would require reintroduction. And, I'd like to pass it over to Annette now to talk about this fact that climate change might actually be one of the challenges that biocontrol faces. And that was the purpose of the research that we've been doing and the focus of this coffee talk today. So with that, I'll stop sharing my screen and let Annette take over.
Thanks, Carrie. Right. So for the next 15 minutes or so, I'm going to go through the management challenge. So if you didn't get a chance yet to read over it, don't worry, you won't be missing out. And we'll also have a link to this in the chat if you would rather see it on your own screen. And so as Carrie mentioned, the purpose of this management challenge was first to synthesize just what evidence there was out there about the impacts of climate change on biocontrol agents and their target hosts. And so as she showed in her presentation, this was really stemmed from feedback from those surveys. So the original Northeast survey in 2016 and the follow-up that we did just this past year, in both of those, climate impacts on biocontrol agents were among the top 3 high priority categories. And there was also a lot of write-in interest on this topic. And so we do have a more systematic review of climate change impacts on biocontrol agents that was just submitted. So keep your fingers crossed that we're successful through that review process, and with any luck that will be out next year and you'll see some more risk-related resources released. But for now, we have this 2-page management challenge. So if you're unaware, this is a resource that RISC puts out. They're usually only 2 pages long, and it's a way for us to synthesize information on topics and present it in a hopefully accessible way. And so the way that our management challenge is structured is that we start off with a bit of an overview, which I'm not going to go into too much since Carrie did a much better and in-depth overview of biological control just now. But really the focus was to talk about how climate change could be complicating the use of biocontrol and kind of adding to some of the challenges that Carrie just mentioned. So as we know, biocontrol agents, to be a successful biocontrol agent and to be approved, they need to have a clear relationship, either ecological and or evolutionary relationship with some kind of target host. So this could be an invasive species, it could be some kind of nuisance species like a mosquito or something that's impacting public health. And we know that there's a couple of different ways in the literature that we're already seeing climate change potentially impacting biocontrol agent programs. And so we've tried to synthesize these different impacts through this figure that I'm sure that hopefully you're seeing on your screens. And we really wanted to emphasize the point that climate change is likely to impact not just the biocontrol agent, which is this blue happy smiley face here to the left, but also since there's this tight relationship between biocontrol agents and their target hosts, we also need to consider how climate change might be impacting these target hosts. So, for example, an invasive plant. And the impact of climate change on these 2 different organisms could result in a variety of different responses and potentially result in a couple of different mismatches. And Carrie alluded to a couple of these in her presentation as well. And so when we were looking through the literature that we turned up through our systematic review, we found that a lot of these responses that were being measured and potential mismatches fell into one of kind of 5 broad groups. So we see potential mismatches in phenological responses. So these are the responses for like timing or onset of activities. So this could be where a species like a plant might leaf out earlier in the spring because of warmer temperatures, and its biocontrol agent, if that's a herbivore, maybe isn't active at that time. So that's an example of a potential mismatch. There could be morphological mismatches. So that's mismatches in the size or shape of organisms, which could be important if you've got a predator targeting a particular host and they can only eat a certain size of host. Another way that there could be climate-related mismatches is in the movement or behavior of organisms. So this is an example of where we might see a target host range expansion that doesn't match the range expansion of its biocontrol agent with climate change. We could also see physiological responses, so things like metabolic rate and feeding efficiency between host and agent. And then lastly, differences in their reproductive cycle or developmental rate. And so this could be important if you've got, for example, a parasitoid biocontrol agent, which targets a particular life stage of a host. So if a particular parasitoid wasp, for example, only lays eggs at a certain life stage of its host, and that host is developing much faster, then that time window for which the biocontrol agent could be efficient is much narrower under climate change. And so these variety of different response mismatches can result in overall changes to the biocontrol process. And so often when biocontrol agents are being considered for approval, we need to prove three key concepts. So we need to prove that that biocontrol agent is able to survive in that introduced range. We also need to prove that they're able to reproduce in that introduced range so that they can form self-sustaining populations, since this management strategy is meant to be a long-term management strategy. And then lastly, we also have to prove that those biocontrol agents are actually efficient at managing that target host. There's no point in releasing something if it's not actually going to do the job that we are releasing it to do. And so these three core kind of steps or components of this biocontrol process could all be influenced by potential climate-related mismatches. And so I want to, for our second page of the management challenge, we kind of dig into particular case studies to show potential climate change impacts for these three different categories. And so for the first category, which is our survival category, the case study that we report in our management challenge is one for the alligator weed flea beetle. So this is a biocontrol agent that has been introduced to control alligator weed, which is a noxious aquatic plant, particularly common in the southeastern United States. So it forms these really dense mats, and it can have a lot of both ecological and human-related impacts. So it impedes the flow of water and can also affect recreational activities. And so in this particular study by Liu et al., they looked at how warming conditions in a field experiment might impact the survival of alligator weed flea beetles and target host alligator weed. And what they found is that when they simulate warming conditions out in the field, that increased the overwintering survival, so the ability of the biocontrol beetles to survive over the wintertime. And this resulted in, this much higher survival resulted in higher beetle populations. And the authors concluded that it also could help facilitate range expansion of these biocontrol beetles, which is great news for the use of alligator weed flea beetles under future climate conditions. But they also found that warming increased the survival of alligator weed, but because beetle herbivory increased at a much faster rate under these warming conditions, the study concluded that at least where these biocontrol beetles still co-occur with alligator weed, that this increased beetle herbivory is likely to enhance the effectiveness of this biocontrol agent under climate change. So, this is one example of where, although we see an increase in survival for both the biocontrol agent and the target host, why it's so important to look at both agent and host together, because again, it's really this relationship between the two that impacts the performance of biocontrol as a management strategy. In the second case study, we're focusing on reproduction. And so in this study, they looked at how heat wave conditions that have been simulated in the lab impact the reproductive performance of a parasitoid wasp called the samurai wasp, which is used to control brown mum-rated stink bugs in the US. And what they found was that heat wave conditions lower the emergence of parasitoids, so slow down the development of these parasitoid insects. And they also slow down and lower the hatching success rate of the target host. But what they did find also is that once those parasitoid wasps have developed, the actual parasitism rate under these heat wave conditions doesn't appear to be affected. And so because the development or reproductive rate for both the biocontrol agent and the target host is being negatively impacted, but the parasitism rates aren't, although there's some potential slight change that we might experience with climate change for this particular case study, again, the biocontrol agent is likely to still be successful where it's co-occurring with this target host. So that's another sort of good news story, we think. And then our last case study is to look at the actual performance of the biocontrol agent and its target host. And so in this case study, they didn't look at temperature. The authors looked at changes in carbon dioxide levels. And so for this case study, they looked at a biocontrol beetle called Gradiana boliviana. It's a really host-specific biocontrol beetle. It only feeds and reproduces on tropical soda apple, which is obviously its target host. And tropical soda apple is a noxious weed that has a variety of agricultural impacts in the United States. And so what the authors did is they used elevated carbon dioxide conditions to simulate future climate change and compared the responses of biocontrol beetles and tropical soda apple to carbon dioxide levels that are representative of today's climate. And what they found is that under high carbon dioxide levels, the biocontrol beetles ate much less leaf area compared to ambient conditions. And this lower feeding rate suggests that they might not be quite as efficient under these high CO2 levels that are predicted for the future. In contrast, the tropical soda apple, which is that target host, they really benefited from high CO2 levels. So they showed much higher biomass growth under elevated carbon dioxide, again, in these growth chamber experiments. And so together that suggests that maybe for this particular biocontrol system, future climate change may mean that there needs to be other additional management supplements to help support the performance of biocontrol agents for managing tropical soda apple. So sort of thinking about exploring other integrated pest management strategies. And then at the bottom of most of our management challenges, if not all of them, we have a variety of management implications. And so, one of the main takeaways that we were hoping people would take from this management challenge is the importance of considering how climate change could impact biocontrol agents and start incorporating that into the agent testing and selection process. And what I've hopefully shown you and convinced you is that it's really important to not just look at the performance of the biocontrol agent, but also to look at how the performance of its host could also vary. And what we've found is that a lot of the studies and a lot of the data that's out there is focused most primarily on the effects of temperature. But as we all know, there's more than just temperature that's likely to be changing with climate change. And so we need to do a lot better job at looking at some other climatic conditions, and the interaction between those. So thinking about how warming, drought, and carbon dioxide might interact to influence survival, reproduction, and performance of agents and hosts. And this is important because depending on what the data is showing for how these climate change impacts, we may need to adjust how we're using biocontrol agents and biocontrol programs are structured. So this could include things like adjusting the timing and frequency in which biocontrol agents are released and where they're being released, and also what populations we're sourcing biocontrol agents from. So maybe we need to be collecting more biocontrol agents from populations in warmer regions of the species range, where individuals might be pre-adapted to future warmer climatic conditions, just as an example. And then again, thinking about this interaction, how biocontrol might also be used in conjunction with other management strategies, so things like chemical or mechanical control, and thinking about ways that we might need to start exploring some of these supplemental activities if data is suggesting that climate change might negatively impact biocontrol performance. And then lastly, I just want to really emphasize the importance of building collaborative networks. So, a lot of the things that I've talked about for these implications involve both time and money, which is already pretty limiting for invasive species management and just management in general. And here, we're really looking for increase in data collection, particularly collecting baseline data, which can be used by climate change studies to look at potential climate impacts. And so one of the ways that we think that this could be addressed is by forming more collaborative relationships with researchers, such as those in RISC, because we often have a lot more capacity to help with field data collection and things like that. So that's one way that we could be getting around some of these time and resource limitations. And one of the other things is that, this is a relatively new foray, at least for me, into this biocontrol world. And so often researchers aren't aware of what taxa people are particularly interested in or are using. And so, help identifying those high priority taxa is another way that these collaborative relationships could help boost our understanding of potential climate impacts on biocontrol. So that was my whirlwind tour through the management challenge. Hopefully you found this useful. I'm going to stop sharing my screen, and we have a couple of minutes for just any general broad questions from the group before we split into breakout rooms and have more of an in-person discussion about your thoughts on the management challenge, thoughts on biocontrol and climate change and such. Rob.
Yeah, so this is really good information. I'm actually thankful that, folks such as yourselves are researching this, and, given the time it takes to research and get biocontrol approved, a piece of that research has to be, how will it perform under different climate conditions 15, 20 years from now. I wanted to just ask a question. I saw a presentation, and Eva, you may have seen this. It was at the NASMA conference, and it was a discussion on chemical ecology and biocontrols specifically. And it was a discussion by a Dr. Park, who was talking about how chemical compounds released by plants could be used to enhance biocontrol by increasing host specificity. Enhancing the chemical attractant to better lure the biocontrol would be similar to training the biocontrol to, more aggressively target, the target species. And I'm just wondering if that might play a role in some of the climatic adaptations when we're talking about, the impacts of the biocontrol and changes with climate. It would appear that there might be some linkage there, and I just thought I would throw that out there.
Yeah, that's a really good point. And I'm trying to think of those 5 categories that we presented on that first page, if it would fit into one of those. I mean, maybe a physiological release of compounds, is it a physiological response? But yeah, no, you're totally right. It's something that we didn't see when we were doing the systematic, like sciencey side of this. We didn't see a lot of data on that. Again, we were specifically looking for papers looking at biocontrol agents, so there might be more of a one-sided story where people have looked at how climate change impacts, the plant, invasive plants, if it's, a plant that's trying to be managed. But yeah, it was really startling, at least to us, about how few studies looked at both species at the same time. But I agree, yeah, it's definitely something that I think is deserving of more research in the future.
I also just wanted to, before we move to our next question, just to welcome, we do have like some biocontrol experts in our audience that I'm very grateful joined this call. So if someone does know the answer to one of the questions or has a comment to share, please feel free to jump in. This is the purpose of the Coffee Talks, is to be interactive. So take it away, Nancy.
Thank you, Carrie. Yeah, I'm fascinated by the suggestion that research on how the agent responds to different parameters associated with climate change could be incorporated into the pre-approval testing process. I would love to hear from folks who have been, nominating agents. I have understood in the past that the process is already very, very slow and can present a barrier for sort of the work. So I'm trying to understand. I would like to hear from someone who has experience with the process, whether asking for additional research as a condition of release is a good idea. I'm agnostic. I don't know. It's certainly, it would mean that the research gets done. And the question is do our colleagues who are on the regulatory end have the capacity that they would need to do that in a timely manner?
Jen, do you want to take that one, since I have no experience in this?
Sure, I can't really speak entirely for this, but I would say I think it's a really excellent thing to add, but I think I would hesitate in calling it a requirement, in part because usually biocontrol programs aren't very well funded, and so finding funds to have that additional research might be complicated. And I also think sometimes it's hard to find the right experts who could help figure out all those details, can be really challenging as well. And so I think it will be valuable, but I wouldn't want to see it become a requirement. And I think we are seeing more and more as petitions are going in, we're seeing more and more information about climate matching. But I think that that is a useful component, but I don't know that it's a required component at this point. So hopefully that answered your question, Nancy.
Thanks, Jen. So Eva, maybe this is a good time for us to split into breakout rooms after we've done Carol's question. Carol,
Well,
take it away.
In addition to what Jen's saying, I think the one thing about biocontrol approval processes is this is not something that's done in a black box. There's an environmental assessment that's done. It's put in the Federal Register. It's open to public comments. And so what we're finding is that, when the public has concerns, they're bringing it up in the EA process, then it ends up getting back in. There's an iterative process to how we're approving these things. So, one thing that's been coming up lately is, people are wanting to make sure that when we are permitting these new biocontrol agents, they're not going to have a negative effect on pollinators. So there's a lot more work that's being done on pollinators on the front end, because when we go through the process of approval, we're being told, this is something we want you to be aware of, and we want to know what's going to happen. And so, that's one thing to keep in mind, that it's not like there's a black box. If there's a legitimate concern that comes up, it does get addressed by the regulators. I'm not sure if that was helpful or not.
I think that was particularly helpful. Thank you.